Hey everybody, and welcome to the first of our lecture videos. This first chapter that we're going to do, or this first section, is on water. We're going to talk about just a little bit of the properties of water and some of the interactions that exist within water molecules. Hopefully by the end of this you'll understand a little bit more about why water molecules are able to dissolve polar or charged species as opposed to nonpolar ones, and then just a little bit of background information about some of the interactions that take place between molecules that are non-covalent. Non-covalent meaning no sharing of electrons. All right, so this is the water molecule down here. Now, water is described as a solvent because of its ability to solvate or dissolve many but not all molecules. Molecules that are ionic or polar dissolve readily in water, but non-polar substances dissolve poorly, if at all. Oil, for example, is a non-polar molecule, and it separates from water when mixed. On the other hand, salt or sodium chloride, which ionizes into ions, right? And ethanol, which is polar, are both able to form, ethanol is able to form hydrogen bonds. Sodium chloride is able to ionize. Both of these mean that it dissolves in water, or they dissolve in water. Ethanol solubility is crucial, obviously, for people who brew beer or who are into making wine, because if it didn't, you wouldn't be able to get, if these two things didn't form a solution, if they didn't mix with each other, then these things wouldn't exist: beer, wine, and other spirits. So we use the term hydrophilic to describe substances that interact well with water and dissolve in water, and the term hydrophobic to refer to materials that are nonpolar and do not dissolve in water. And so in this table here, hydrophilic versus hydrophobic, you can see a couple of the representatives of these substances. There's a third term called amphiphilic, which refers to compounds that have both properties. So soaps, for example, and I'll show you a structure of soap here in a little bit, are amphiphilic. They have long nonpolar tails, and they have these shorter polar heads, but essentially they have groups belonging to both of these categories. Now, the solubility of materials in water is based on what we would call free energy changes, and it's measured by a term delta G. Some of you from chemistry might remember delta G and its equation. We have delta H, which is enthalpy, and I believe this gives us a negative T delta S. I think this was the equation. It's in our book. We can take a look at it a little later too. H is enthalpy, S is entropy. So if T is in Kelvin, and we're talking about things that are spontaneous, things that are favorable in as far as reacting or reaction-wise in the forward direction, delta G has to be less than 0. It needs to be a minus number. It needs to be negative, less than 0. And so from this equation that we can see that lowered delta G values will be favored, or to get them lower, obviously decreases in enthalpy and or increases in entropy. These are things that will favor the delta G. Let's see here. Let's consider why nonpolar materials don't dissolve in water. So we could imagine a situation where the process of dissolving involves the surrounding of each molecule of the nonpolar substance, whatever it is, in water, just like sodium chloride gets surrounded by water molecules as the salt dissolves. There's a significant difference between the surrounding of a nonpolar molecule with water, as in like surrounding ions or polar compounds with water. The big difference is that since nonpolar molecules don't interact with water readily, the water behaves differently than it does with the ions or the molecules that form hydrogen bonds, like ethanol. In fact, around each nonpolar molecule, water gets very organized. It aligns itself regularly, and if you remember anything from Gen Chem about entropy, that entropy is a measure of disorder. So when things become more ordered, entropy decreases. And in our equation, negative of a negative delta S, right? If this term right here starts to go down and we have a negative already in here, well, that's going to be a positive. So a positive value would not be good for spontaneous. And so this is, again, we're talking about why nonpolar substances don't readily dissolve in water. Well, it's because the entropy of the water tends to increase. The water gets more ordered. We're going to look at how water interacts with itself here in a second. So since the mixing of water also doesn't really have any significant heat component, delta G is going to end up being positive in this example. This means then that dissolving a nonpolar compound in water is not favorable. So when we talk about nonpolar material associating with itself and not water, it's really that the water associates with itself, it's free to mix, and then it organizes into a very ordered structure, kind of pushing out all the nonpolar materials. Okay, so when things actually do dissolve, we get some variations in that enthalpy and that entropy. Obviously, that usually substances, when they dissolve, they tend to give off some heat, and giving off heat, if you remember from enthalpy, that's a negative value. So a negative delta H tends to help negative delta G. Okay. So let's talk about the amphiphilic substances. So amphiphilic substances like soap. I had mentioned soap. So let's talk about what happens when we mix amphiphilic substances with water. So here you have a soap molecule, this COO2. If you remember our organic chemistry or just our Lewis structures, this is actually, so we can kind of erase that here. We've got a carbonyl carbon, C double bond O, and then we've got a negative on this charge here, on this oxygen here, because this is the carboxylate ion of our salt, of our acid. This is a fatty acid, to be specific. So the H is missing, so it's the salt of the acid, and it's associated with an ion, a positively charged metal ion. So the sodium ion's attached to the fatty acid, and it comes off really easily in soap when we put this in water, leaving behind a negatively charged molecule at one end and a nonpolar region at the other. And so the ionization of soap causes an increase in entropy, the two particles instead of one. Increase in entropy, of course, meaning more disorder. The nonpolar portion of the negatively charged soap is the problematic part. If it's exposed to water, it will cause water to organize and result in a decrease of entropy and a positive delta G. So since we know fatty acids dissolve in water, there's got to be something else at work here, because like we said, if we get a decrease in entropy, that leads to a positive delta G, at least in terms of our equation. So there's something else going on, and just like nonpolar molecules in the first example that we looked at, associating with each other and not with water, so too do the nonpolar portions of the soap ion associate with each other and exclude water, and the result is that the soap ions arrange themselves into something called micelles. And so I've got a little picture here of micelles as well. So the nonpolar portions go on the inside of the structure, away from the water, and the polar portions stay working on the outside with the water. And so these interactions of the polar head groups with the water returns the water to its more disordered state. Remember, if the water has nothing else to interact with but itself, it's going to form these really highly organized hydrogen bonds with each other and form these little lattices, and that's an increase in order, which is a decrease in entropy, which is going to, because entropy is disorder, remember, so an increase in order is a decrease in entropy, which is going to lead to a positive delta G in our equation. So by allowing water to interact with these groups, as opposed to not interacting with them, we get less of that happening, and so our delta G is going to stay negative in terms of when things actually dissolve. So this increase in disorder drives the formation of these micelles, and as we'll see later when we talk about lipid bilayers, The same forces drive glycerophospholipids and sphingolipids to spontaneously form bilayers, where nonpolar portions of the molecule interact with each other to exclude water, and the polar portions arrange themselves together on the outside of the bilayer. It's pretty neat. So this is how these things are able to actually dissolve in water, things like soap. And these micelles actually do form in soap. Bilayers are a little more involved, right? So you can see the micelle is just a single layer, polar groups on the outside, non-polar groups facing inward. When we expand this to be the outside of our cells, we're going to see that by having a bilayer, we can have aqueous environments on both the outside and the inside layers of our cells, but while keeping them separate. Okay. So let's talk about, let's see, I'm going to tell you what this slide is here. Okay, so interactions of the, so again, those bilayers that we're talking about, this is just a better picture of that bilayer that I was just going over right now. Again, this is maybe the inside of the cell, maybe this is the outside of the cell. You can see here all of the non-polar portions are facing inward, dehydrated implying that there's no water, semi-saturated, the kind of interface in between the outside aqueous environment and the inside non-polar environment, is going to have some associations with water, and then you can see the polar groups all the way on the outside. Now this other image that I'm showing you is a little prequel of what we're going to see when we get to protein folding. When globular proteins actually fold in the cytoplasm, the non-polar amino acids are found in the interior portion of the protein, and the polar amino acids end up being on the outside. And so they exclude water on the inside by interacting with each other, all the nonpolars, and the interaction of those amino acids turns out to be one of the driving forces for the folding of proteins as they're being made in aqueous solution. So here the yellow represents the polar and the black represents the nonpolar, and so you can see the sort of segregation here of this protein's different groups as it folds. All right, so let's talk about hydrogen bonding. It's hard to overstate the importance of hydrogen bonding, as Linus Pauling once said. Actually, I'm going to show you another picture too. This picture that I have here is just showing us some of the basics. So let's talk about some of the basics here real quick. So when we talk about hydrogen bonding, we're talking about hydrogen being attached to an oxygen, a fluorine, or a nitrogen directly. Now there can be other things on those other atoms, but it's the H's that we're interested in. And so when they are bonded to, hydrogens are bonded to these three atoms, there's a very uneven sharing of electrons. So we get some partial charges. We're going to talk about that in a minute. And so it's a weak bond. What happens though is that those hydrogens end up being donors that an acceptor can actually kind of pull on. And so we'll see other electronegative atoms, such as nitrogens and oxygens, can actually come in, and if they get near hydrogens that are bonded to other electronegative atoms, there will be an attraction between them. And so hydrogen bonds can exist between hydroxide groups in water, between oxygens of water and a hydrogen of a different group, right? So we can even exist from the proton in water to the oxygen of a different water, or a carbonyl group. These C double bond O's are going to be also participating in hydrogen bonding. So there's a lot of hydrogen bonding going on. Let's talk about partial charges. So the difference in electronegativity between hydrogen and the molecule to which it's covalently bound gives rise to partial charges. These tiny charges, often indicated or shown as deltas, so delta plus or delta minus, meaning partial, result in the formation of hydrogen bonds, which occur when the partial positive charge of a hydrogen atom is attracted to the partial negative of another molecule. So in water, that means hydrogen of one molecule is attracted to the oxygen of another. And since water is an asymmetrical molecule, and here you can see quite a few of those waters, it also means that the charges are asymmetrical. So such an even distribution is what such an uneven distribution, sorry, is what makes a dipole. So dipolar molecules are important for interactions with other dipolar molecules and for dissolving ionic substances. And so you can see a few of those shown here. Hydrogen bonds are not exclusive to water. In fact, they're very important forces that hold together lots of macromolecules, including proteins and nucleic acids. Hydrogen bonds do occur within and between macromolecules. So here we're looking at just a couple of interactions, ion-dipole interactions. So here you can see an ion in terms of K plus interacting with the dipole nature of some of these water molecules, or the anion ion again reacting or interacting, sorry, with the partial charges of a dipole. You can see dipoles reacting or interacting with each other as well. So partial, partial positives here of water with partial negatives of the oxygens, and partial negatives of carbonyls with partial positives of, other carbonyls such as the C double bond O of another group. So all of these partial charges can interact with each other. Complementary pairing that occurs between opposite strands of DNA is also another example of hydrogen bonds. Each hydrogen bond is relatively weak compared to a covalent bond, for example, but collectively they can be pretty strong. So just to take a look at that, here's a covalent bond energy. So energy, of course, being the energy we'd have to put in to break these bonds. So you can see here in the 400s of kilojoules per mole just to break a hydrogen carbon bond or an oxygen hydrogen bond. But hydrogen bonds that we're talking about here, or ion dipoles, or even those hydrophobic interactions, are really weak, and so that's why we call these weak forces or weak interactions. The bond energy is a fraction of that. All right, that's not a bad thing though, because their weakness is actually quite beneficial for cells, particularly as regarding nucleic acids. Strands of DNA, for example, must separate over short stretches in the process of replication and in the synthesis of RNA. Since only a few base pairs at a time need to be separated, the energy required to do this is small, and the enzymes involved in the process can readily take them apart as needed. Hydrogen bonds also play a role in the binding of substrates to enzymes, catalysis, and protein-protein interactions, as well as other kinds of binding, such as protein DNA or antibody antigen binding. As noted earlier, the hydrogen bonds are weaker than covalent bonds, and their strength varies from very weak, like 1 to 2 kilojoules per mole, to fairly strong, 29 kilojoules per mole. Hydrogen bonds only occur over relatively short distances, like 2.2 to 4 angstroms, 4 angstroms being 4 times 10 to the minus 10. The farther apart the hydrogen bond distance, the weaker the bond is. The strength of the bond in kilojoules per mole represents the amount of heat that must be put into the system to break the bond. The larger the number, the greater the strength of the bond. Hydrogen bonds are readily broken using heat. Boiling water, for example, requires breaking hydrogen bonds. When a biological structure, such as a protein or a DNA molecule, is stabilized by hydrogen bonds, then breaking those bonds destabilizes the structure and can denature the substance, which is losing of structure, or denaturing is the loss of structure. It's partly for this reason that most proteins and all DNAs lose their native or folded structures when they're heated or when they're boiled. For most DNA molecules, denaturation results in a complete separation of strands. For most protein, this means their characteristic 3D shape is lost, and usually the function along with it. Some proteins can readily reassume their original structure when the solutions that they are in are cooled, but most can't. This is one of the reasons that we cook our food. Proteins are essential for life, So denaturation of bacterial proteins results in the death of any microorganism contaminating our food. And again, all of this is possible, right? Heating our food and killing the bacteria is possible because of the weak interactions that we see here. And so we're going to go in more in detail into what these structures are. If you recognize guanine and cytosine by name, you know this is DNA and these are base pairs. But that should do it for this first lecture on water. Stay tuned for the next lecture in the series. All right, guys. See you next time.